our first speaker, Regine Claßen-Bockhoff, who will be presenting on Syncephalia development in COPS.
Where is the presentation?
I'm sorry, what was that?
You'll need to share it like you did before. Share your screen.
I'll do it again. Okay.
Yeah.
So now you see my presentation?
We do, yes.
Okay. And you also see the cursor?
Yes,
we do.
Okay, that's fine. Okay, so yeah, thank you for the invitation and thank you for listening to my talk. I'm Regine Claßen-Bockhoff and I'm interested in the biology of inflorescences and particularly in the formation of flower-like patterns in Pseudanthia. And Asteraceae is a fantastic family to study this topic, because we have capitula and clusters of capitula called Syncephalia, which are both interpreted as inflorescences. And today I will talk about the convergent evolution of Syncephalia together with my co-authors. Jasmin Wahl and Dieter Langerbeins are former students of mine from Mainz University and Aachen University, and Jakub Baczynski is a colleague from Warsaw University. So the term Syncephalium is not precisely defined. In the past, it was called secondary head, double head, glomerule, pseudocephalium, cephalodia, or Syncephalium. And we use the term Syncephalium in the sense of Kunze and Harris as a superordinate term for all these forms of aggregated capitula. And Harris estimated that about 63 genera form dense capitular clusters, and among them 30 genera from the Gnaphalieae, which is the group with clearly the most Syncephalous genera. In my talk, I asked the question, are Syncephalia inflorescences? Though they are usually interpreted as inflorescences or inflorescences, and it is usually said that they flower in a centrifugal or basipetal direction. But these assumptions have never been tested. This study continues our previous study in which we investigated capitula and asked, are capitula inflorescences? And these capitula are usually interpreted as condensed inflorescences, but developmentally they turned out to be floral units developing from flower-like meristems rather than from inflorescence meristems. Let's start with the floral unit meristems. They are determinate. That means that they have lost the capacity of terminal growth, of apical growth. They have no central zone or visual clavata system which continues open growth. They are determinate, and they are able to enlarge before and during subdivision. And the process of subdivision of such a determinate meristem is called fractionation. This is the process which completely uses a determinate meristems and fractionates subunits. So in a flower meristem, these subunits are floral organ primordia. In the capitulum meristem, the subunits are flower meristems, which will fractionate floral organ primordia in the second step of fractionation. And in more complex units, we have capitular meristems, which will fractionate flower meristems, which will fractionate floral organ primordia. So these three units differ in the number of fractionation steps and share most of the morphological and developmental characters, and as far as known, they also share epigenetic and genetic regulation mechanisms. We call this complex unit, a caput meristem, to distinguish it from an inflorescence meristem. So the caput meristem is the first way to produce Syncephalia, which are rare in Asteraceae, and only two genera are examined in more detail. These are Pycnosurus and Echinops. So both species, which are illustrated here, start with a large globular floral unit meristem, which fractionates in capitular meristems, which fractionate into flower meristems. And in Pycnosurus, we see that this fractionation is in acropetal direction. We have these drop-like meristems, which split into an abaxial scale and an adaxial capitulum meristem. The capitular meristems develop flower meristems, and during this process, the whole syncephalium enlarges and reaches such a globular shape. Flowering sequence is almost simultaneous with a weak centripetal gradient, which you can also see here is a little bit more developed in the base than in the top. In Echinops, the capitular meristems have no scales. They are initiated in a naked mode, and the capitular meristem produces up to 20 spiny involucral bracts, just seen here, and then the capitular meristem directly transitions into a flower meristem, resulting in one-flowered capitula. So this is interesting because the capitulum meristem, if the second fractionation step fails, it transitions into a flower meristem. During development, the unit gets an inverse pear-shaped shape and has a apical zone which is more promoted than the basal zone. And interestingly, this syncephalium flowers in basipetal direction. And it is assumed that this is due to spatial constraints, that in the apical region there's more space, that the primordia and the meristems are promoted compared to the proximal ones, and that this may be correlated with the basipetal gradient. But this has to be checked with statistical methods in a future study. So we have seen that caput meristems can form Syncephalia, and with this little table, I like to explain the different meristem types. So the flower meristem is determinate. It has no stem cell activity and can expand during fractionation. Histologically, it has a mantle meristem, and the process of subdivision is fractionation, with the result that floral structures appear. The floral unit meristem, which is a capitular meristem, shares the same meristem features, but differs in repeated fractionation. So the first step produces flower meristems, which then fractionate floral structures. In contrast, inflorescence meristems share more characters with shoot apical meristems. They are both open, have a stem cell activity, they have a central zone with peripheral meristems, and continuously segregate lateral meristem parts, which will develop in bracts and flowers or in leaves. The inflorescence meristem differs from the shoot meristem in its limited activity and in the axillary meristems, which are always reproductive and develop immediately, whereas in shoot meristems, they develop with delay. Now we continue with Syncephalia formation on the inflorescence level. And our first example is Ligularia stenocephala, which has a terminal syncephalium composed of lateral capitula. A terminal unit is lacking, flowering sequence is acropetal. When we look at the developmental pictures here, we see the inflorescence meristem, which is still in the process of segregating bracts and axillary meristems. These axillary meristems develop floral unit meristems, capitulum meristems, which then fractionate into 1, 2, 3, 4, 5, and a sixth flower meristem is covered here. And only one flower meristem develops a ray flower. This is very interesting in this species. Within the genus, we have one species, Ligularia sibirica, which has a terminal capitulum. And you can see that the flowering sequence, the development sequence is acropetal, but the terminal unit is larger and promoted compared to the distal ones. So the terminal capitulum belongs to the main axis, whereas the lateral capitula belong to branches of first order. So we see that flowering and development follows the branch order, that this one is in advance compared to the lateral ones. And this is a general feature in closed inflorescences, that the terminal unit, irrespective of whether it's a flower or a capitulum or an inflorescence, always usually develops in advance compared to the distal ones. And this flowering after the branch order leads us to ordinal flowering in Polycaluma. This is a desert plant from Australia, which very complex Syncephalia clusters. So we have in the young stage, you see that the Pseudantium is composed of many up to 100 capitula, which apparently flower in a centrifugal pattern. In the later stage, you cannot identify the individual capitula anymore. And if you analyze this pattern, you see that it is a pliocasium. We have a terminal capitulum, and in this case, we have 8 cymes, which develop in a dicasial, rarely tricasial or monocasial pattern up to the 5th branch order. And with each branching, the capitula get a higher branch order. And this is also reflected in the number of flowers per capitulum. The terminal unit is the largest one. And here, this is the 4th order, has only few flowers. So we have an ordinal gradient. And what looks like a centrifugal pattern here is in fact an ordinal pattern, because we have always a change of the branch order with each after each capitulum. Polycaluma starts with an inflorescence meristem. Very early, we have the terminal capitulum meristem, and in this case, 8 lateral cyme meristems. In this stage, you may argue that it looks very much like a floral unit meristem, but if you look at the lateral meristems, you see that these are cyme meristems, which continue branching. So the terminal capitulum develops the first, and then we have the lateral meristems, which each produce a capitulum of first order and 2 prefills, and in the axils of the bracts, cyme meristems, which repeat this process until the 5th order. And here in this older stage, you see the characteristic zigzag pattern of cymose branching, which indicates that we have inflorescence, a pliocasium with cymose branching patterns. Let's stay a short moment with Pseudantia. The most important or the most known example is the European edelweiss, which is a botryoid or rarely a reduced panicle, surrounded by bracts which are densely haired with woolly hairs And this is obviously an adaptation to the alpine habitat. And the same pattern is found in the New Zealand Edelweiss, where you have a botryoid with these enlarged subtending bracts, again with these woolly hairs. These are, let's say, simple aggregations, but more spectacular are the pseudanthia in the close relationship of Polycalyma. Here you see Amoebium alatum, which is a simple capitulum. You have showy involucral bracts, you have no terminal unit, and you have a centripetal flowering sequence, so a typical capitulum. This is Myriopcephalus helichrysum, which looks very much the same, but flowers in centrifugal direction. And the morphological analysis reveals that we have a terminal unit and lateral capitula, and these capitula are composed of 3 flowers. So this unit reveals as a caput with 3 flowered capitula, a terminal one and lateral ones, all sitting on a secondary receptacle. Unfortunately, we don't have developmental data so that we cannot interpret this centrifugal pattern, whether it is cryptic cyme or raceme or just the effect of spatial constraints. We cannot, we don't know this yet. And this is Gilberta tenuifolia, a very small pseudanthium, only half a centimeter in diameter. And this has bracts, it has no terminal unit, it has centripetal flowering, so looks very much like a simple capitulum. But if you look at the morphology, you see that the outermost flowers are surrounded by some additional bracts, which it's interpreted that these are one-flowered capitula. So this unit is interpreted as a caput composed of one-flowered capitula, and you see in the center these bracts, additional bracts are lacking, and this system transitions into a capitulum. So what we saw in Echinops, that a capitulum transitions into a flower, is repeated here, that a caput is transitioned into a capitulum in the center. This indicates that these units, flower, capitulum, and caput, are very similar and distinguished by these steps of fractionation. There are only 2 genera known with complex pseudanthia with ray flowers. This is the Mexican Dyssodia decipiens and the South African Oedera capensis. And both are botryoids with terminal capitulum and lateral capitula, and only these have ray flowers. So this is artificially separated to show the organization of this pseudanthium. In Oedera, we found one blossom with an elevated terminal capitulum, and interestingly, this terminal capitulum forms 13 ray flowers. And in Dyssodia, we also found some anomalous forms, and they showed ray flowers around lateral capitula if they are elevated, or at the terminal capitulum if lateral capitula are inhibited. So this this indicates that we have a different regulation compared to a simple capitulum. In a simple capitulum, you have a centripetal gradient which determines the flower morphs. But here it appears as if mechanical pressure would inhibit the development of ray flowers, that the capitula have the potential to form ray flowers all around, but that the innermost are maybe inhibited by this dense aggregation. So now we go back to Syncephalium organization. And here we have an example of a tertiary syncephalium. So in Asteraceae have many, many inflorescence forms, simple ones and very complex ones. And Harris distinguished the capitulum, secondary and tertiary syncephalia. And she mentioned this example and argued that all these tertiary syncephalia will flower in a basic petal direction. However, when we look at the morphology of this system, we see that the flowering sequence is acopetal. Here you have young developing syncephalium, and you see that the proximal units are more developed than the distal ones. So this is an acopetal development. And you can also see it here on the diagram, which figures the whole syncephalium composed of many subunits, secondary units. And the white circles are flowers in a flowering stage, the gray ones in a prefloral stage, and black ones in bud stage. And you can see that the development and flowering sequence is acopetal, except the terminal unit, which again develops in advance. It is larger than the distal units, and it is more developed, further developed. So we have the same pattern as we saw in Ligularia sibirica. And if we look at this subunit, we have a terminal flower surrounded by usually 5 lateral flowers, and each flower is surrounded by involucral bracts. So this unit can be interpreted as a botryoid with 1 flowered capitula. And looking at this development, we see that the terminal flower again develops in advance, and that the lateral ones flower in an acopetal order. So we have altogether a panicle composed of botryoids with 1 flowered capitula. The tertiary syncephalium flowers in an acopetal direction with an advanced terminal unit, and this is repeated in the secondary units where the terminal flower also flowers before the terminal ones, the lateral ones. So it's a bit complicated. So this system flowers is a mixture of acopetal and advanced flowering of the terminal capitulum. But when we look at the entire plant, this is also Gundelia tonfortii. This is the entire plant, and each circle is 1 tertiary head. Then we see that the tertiary heads flower in acopetal direction, but all the lateral branches flower in a basipetal direction. The symbols are again buds, prefloral, and flowering stages. So, and this pattern can also be interpreted as a panicle, but it is not an inflorescence, but it is a flowering shoot system. And when Harris and Cronquist and Troll said that the inflorescences in Asteraceae flower in a basipetal manner, they referred to the flowering shoot system and not to the inflorescence. They did not distinguish these 2 different reference systems. And because the branching patterns can be the same, but the development is different. The inflorescence develops from an inflorescence meristem, and the lateral shoots from vegetative meristems, which also show here a vegetative base and then become flowering. Schwirb and Stauffer were the first who considered the flowering sequence as a tool for identifying inflorescences, and Hempel and Feldman later confirmed their view by physiological experiments with Arabidopsis thaliana. So irrespective of whether we have a flower, a capitulum, or such a tertiary capitulum. This pattern is very often or generally found in flowering shoot systems, that the terminal unit flowers and develops first, and then the lateral branches develop and flower in a basipetal direction. Interestingly, the shoot system develops in an acropetal direction. So the buds or the axillary meristems are produced in an acropetal direction, but the flowering stimulus shows a basipetal direction, and it is assumed that that the vegetative buds are stimulated by a signal from the terminal capitulum, and this includes two steps. The first step is that the previously or the pre-existing vegetative meristem is activated through decrease of apical dominance. And the second step is that this meristem transitions from the vegetative to the floral unit, to the reproductive stage. And as far as known, auxine signals are involved in both processes. So it is assumed that if the shoot apical meristem become reproductive, that then an auxine flow is directed in basipetal direction, which activates gradually the pre-existing buds and stimulate them to flower. Now, when you imagine that also these capitula of a flowering shoot system aggregate, we come to the third mode of a Syncephalium production, and this is illustrated by Liatris spicata. We have a terminal flowering unit which has a terminal capitulum and many lateral capitula, which however flower in a basipetal direction. And if you look at the development, you can nicely see that in the beginning we have a typical shoot development. We have a shoot apical meristem which segregates leaf primordia, and only in a distance from the shoot apical meristem we see the axillary meristems, which are produced in acropetal direction. So this is a typical vegetative development. But when the shoot apical meristem has transitioned into a terminal capitulum, then you see that these axillary meristems are stimulated to flower in a basipetal direction, and you can see all developmental stages: capitulum development, capitulum meristems, which are floral unit meristems, and vegetative meristems. So in the beginning, the vegetative meristems produce two prefolds and persist in this stage. Then they get the stimulus and transition into a capitulum meristem, which forms involucral bracts and fractionates flowers. So what is the main result of our study? We have seen that Syncephalia evolved convergently from flowering shoot meristems, from inflorescence meristems, and from floral unit meristems. And the processes involved are the extension of the flowering impulse in vegetative systems, resulting in a basipetal flowering sequence, diverse forms of aggregation leading to different kinds of inflorescence clusters, which always flower in acropetal and ordinal directions, and the repeated fractionation, which in a determinate meristem, which leads to a caput with variable flowering sequences. We have also seen that the branching pattern alone may be misleading. Remember the pattern in Gundelia. We have two panicle-like structures, but the one is an inflorescence and the other one is a flowering shoot system. But that flowering sequences and initiation sequences, they refer to organization, and they can be used to identify inflorescences within the flowering shoot system. And we have also seen that the floral unit meristems provide an evolutionary pathway beyond inflorescence aggregation. increasing steps of fractionation lead to more complex units, but also the other way is possible, that fractionation steps fail and the complexity is reduced. So this is a very um, flexible evolutionary mode to form complex units. Inflorescences and caput meristems are also able to form pseudanthia, so we have pattern repetition on different organizational levels. Our study only includes few examples, and Asteraceae have many species, so there are, of course, many other and more modified forms. But it is important for future studies to have in mind that we have these convergent forms, that Syncephalia can arise from different meristems, and that we have some hidden convergencies among the diversity. And this is especially important for evolutionary studies aiming to reconstruct character evolution. Then you must know what the character is. Thank you very much.
Thank you so much. That was a wonderful talk. If anyone has any questions, you're welcome to unmute yourself and ask a question. And you can raise your hand if you'd like to go. Or if not, then you can also put it in the chat and I'll be able to read that. So does anyone have any questions?
Sorry, I have one, but I'm unable to raise my hand. So I'm
Okay,
just
you're
thinking.
good. You can just ask. Yeah.
Okay. Regina, thanks for an absolutely fascinating talk. I actually have about 20 questions for you. I'll try and restrict myself. That's really amazing and fascinating work. I work on Gnaphalieae, mainly from South Africa, so I'm familiar with a lot of these structures, but I've never really had an understanding of them. And of course, you've shed so much light on homology for taxonomic characters here. So I've often wondered about Paley and the homology of Paley with involucrobracts.
Yeah.
Whether there's perhaps hidden Syncephalia with reduction in floral numbers, or yeah, I mean, there's a genus Underbergia where what are considered paley have an unusual structure with an expanded limb, which is identical to the involucrobracts and very unusual, just as an example. So yeah, I'd be keen to hear what you think of the homology.
Yeah, this is a difficult question, and it has been discussed that these are homologues with involucrobracts, or that they only are scales, that they are effigurations from the receptacle. And my personal view is that these are no bracts but effigurations, because we see in the capitulum development that the involucral bracts do not arrive below the capitulum, but they are part of the capitulum. So they are not, they should not be homologized with leaves, but with elements of the capitulum. And we also see that these structures often come from such a drop-like meristem, which then splits into an abaxial scale and an adaxial flower or capitulum, so that they have a common origin. And it is not so that we have, like as in the vegetative part, that we have a leaf primordium, which then forms an axillary meristems, but they arise from a common meristem.
Okay, thank you very much. Okay, so you're saying the paley are outgrowths. They're not, you'd not consider them homologous to
Yeah,
involucrobracts in general.
yes,
Yeah,
I
okay.
would say.
Okay, thank you.
Any other last minute questions? Well, thank you so much. So now we will go to our second speaker of today, Zaynab Sheik, or is it Shaik? I'm sorry, I'm not sure, but we'll go to our second speaker who's also going to be talking about capitulum development.
Thank you.
Mhm.
Thanks to the TICA organizers for having me speak. Okay. So today I'm talking about a centuries-old question in Asteraceae around Gynomonoecy and its repeated evolution, its frequency in Asteraceae. And we're looking into a comparatively new theory around selective advantages conferred by Gynomonoecy, specifically centering on changes in capitulum architecture. And so to look into this theory, we're using the South African species of Helichrysum. There are 245 species in South Africa. And so this work is in collaboration with my two former supervisors, Dr. Nicola Berg, who's just been speaking, Professor Anthony Verboom. Both are based at the Gothenburg Botanic Garden and the Gothenburg-University. And then more recently, Carme, who's on the organizing committee for TICA, I believe, has come on board as a co-author. She's been producing molecular phylogenies for Helichrysum and its relatives over the last few years, so she's really a welcome addition to the team. But by way of background, if we look at sexual systems evolution in Asteraceae, we have convincing evidence that the ancestor to Asteraceae was probably hermaphroditic, and we base that on ancestral state reconstructions and also by comparing outgroups with the basal subfamily Bondidosoideae. But this alternative sexual system, Gynomonoecy, where the capitula contain female and bisexual flowers, is very common. It's the second most common sexual system in the family after hermaphroditism. So this is the typical arrangement in many members of Asteraceae, where you have disc flowers that are bisexual housed in the center of the capitulum, surrounded by these ray flowers, pistillate flowers, which are often furnished with this attractive petaloid ligule. So Gynomonoecy is the second most common sexual system in Asteraceae, but it also has multiple independent origins across the family. So it's evolved multiple times and then been maintained multiple times. And this really raises questions around the evolutionary significance of this sexual system. What are the selective advantages conferred by this combination of pistillate and bisexual flowers? So one of the earliest theories was put forth by someone called Christian Conrad Sprengel, and he hypothesized that these attractive petaloid rays of Asteraceae are important in attracting pollinating insects and thereby promoting cross-pollination. And this is something Darwin actually writes about in The Origin. He describes Sprengel's work as highly probable. So we have some empirical data supporting this theory, i.e. supporting the idea that capitula with attractive petaloid ligules are indeed more frequently visited by pollinating insects. But this is not always the case. So lineages lacking these attractive petaloid rays are visited, in some lineages, they're visited at the same rate or even more frequently. So this explanation applies to some composite lineages, but not all. Another theory is that having these two distinct floret types, florets of distinct sex, makes possible heteromorphic fruits. So this is an example from Dimorphotheca sinuata here in South Africa. And you can see that the fruits originating from florets of distinct sex look very different. Those of the pistillate flowers are quite small, and those of the hermaphrodite flowers, so these are fruits from the same capitulum, but those of the hermaphrodite florets are much larger and are furnished with wings. So this seems quite compelling, but we know of several composite genera where heteromorphic fruits are produced from all pistillate flowers or from all bisexual flowers. And so floral sex differences are not a necessary precondition for heteromorphism in the fruits. So similar to the first theory I presented, this explanation appears to apply to some lineages, but not others. And there are a number of other theories here that I haven't spoken about, like Gynomonoecy as a strategy for greater flexibility in resource allocation to male versus female reproduction, and also theories around reduced herbivory in pistillate flowers, given that they don't present attractive pollen. But the same applies there. For those alternative theories, those explanations apply to some daisy lineages, but not others. So another way to look at this is that Gynomonoecy may confer selective advantages instead by influencing resource allocation at the level of the capitulum and by influencing capitulum architecture. So this is not novel. This is work that was published originally by Ruben Torres and colleagues in 2019. And they looked at capitulum architecture in 43 hermaphroditic and 28 gynomonoecious species across many genera and tribes. So this was a pretty small sample size considering the size of Asteraceae as a whole. And then they looked at capitulum architecture accounting for phylogenetic relatedness. And even with the small sample size, they were able to uncover some interesting patterns. So what they found is that gynomonoecious species have more flowers per capitulum, more flowers per unit capitulum size, and they also have smaller achenes than hermaphroditic species. And so what we were curious about was, are these patterns evident at shallower phylogenetic scales, like among members of the same genus? So this is where Helichrysum comes in. It's a large and morphologically very variable genus. Contains between 500 and 600 species. There are just a handful of genera in Asteraceae that are just that are this large. So this is really an ideal focal genus. And about 245 of the species occur in South Africa. So based on ancestral state reconstructions, Southern Africa, or more specifically the Cape Floristic region, emerge as the origin, the geographic origin of this group. And what's also really nice about Helichrysum is that it exhibits hermaphroditism and gynomonoecy. So about 2/3 of the species in South Africa are hermaphroditic and the remainder are gynomonoecious. The achenes of Helichrysum are always small and monomorphic. So if you look at achenes from female versus hermaphrodite flowers, it's impossible to tell them apart. They look completely identical. And the pistillate flowers are tubular or filiform, never ligulate. So they never have this attractive petaloid ligule. And instead, attraction is performed by one or several rows of colored papery involucral bracts. And so just looking at these capitula, it's impossible to tell which are gynomonoecious and which are hermaphroditic. In this particular figure, A, B, and C are fully hermaphroditic, and D, E, and F are gynomonoecious. But it's impossible to tell the presence of pistillate flowers in the gynomonoecious species. They look like hermaphrodite flowers. So for this piece of work, we didn't gather any new data. We made use of very useful existing data sets, the first of which was this maximum likelihood tree that Carme and her colleagues assembled last year. This was based on 927 COS2 derived multiple sequence alignments, and this was a much bigger tree, but after having pruned out all the non-South African Helichrysum species, we ended up with a tree with 195 tips. Another really important source of data was a treatment of Helichrysum by a now deceased South African botanist and taxonomist, Olive Mary Hillard. She meticulously counted the numbers of female and bisexual flowers for multiple capitula in each of the 245 species of Helichrysum in South Africa. So this was a wonderful storehouse of data. And she also recorded the sizes of the capitulum and the size of the receptacle and the size of the achenes. And so using these two sources of data together, we first performed an ancestral sexual system reconstruction. We assigned sexual systems here to the tips using Hillard's floret counts. And what we see from this analysis is that we have pretty strong marginal probability support for the ancestor to the South African Helichrysum having been hermaphroditic. So the support here is about 0.79. If we look at the number of shifts from ancestral hermaphroditism to descendant Gynomonoecy, excluding the tip, so just looking at the internal nodes, we see at least 9 such shifts. So this is remarkably plastic across this group, transitions between sexual systems. And then we see a concentration of polymorphism in sexual expression among the early diverged species. So I haven't yet spoken about polymorphic species, which are presented here in purple. These are species which are sometimes hermaphroditic and sometimes gynomonoecious. So the pistillate flowers can be present or absent within a given species. And those species are here among the early diverged lineages. And so what this tells us is that plasticity in sexual expression is more or less concentrated among the early diverged lineages. Another thing we can look at is the floral packing density. So here we're looking at the number of flowers per unit capitulum diameter. And you can see that there are 2 exclusively gynomonoecious clades that have unusually high flower packing density. And then if we look at capitulum diameter by itself, we again see this clade emerging as having particularly broad receptacles. So these are large-headed gynomonoecious species. But another way to look at these data is a phylogenetically corrected linear regression. So you'll see here that the x-axis is the log capitulum diameter, and then the y-axis is the number of flowers. And here we're looking at phylogenetically corrected lines. The uncorrected lines are stippled, solid lines are corrected. And you can see that hermaphroditic species and gynomonoecious species both show a positive relationship as might be expected. The slopes of the lines do not differ significantly, but their intercepts do. So what this tells us is that gynomonoecious species are packing many more flowers into a capitulum of a given size. And in unlogged numbers, this is equivalent to about a 3 times difference. This is really remarkable. Gynomonoecious species are packing, on average, 3 times more flowers into a capitulum of a given diameter. Now, our initial thought was that gynomonoecious species are achieving this by packing in very many more pistillate flowers at their periphery. The reason being that pistillate flowers lacking andresia are skinnier, they're thinner. But what we found was not that. So here again, you have the same figure, but the y-axis now is the number of bisexual flowers. And it turns out that gynomonoecious species are packing many more flowers into their capitula, and they're achieving this by adding very many more bisexual flowers. So this was rather unexpected. And what we suspect is happening here is that the flower numbers are allometrically controlled. So if you look at the radius of a circle, if we approximate the receptacle to a circle, which is the case from looking at herbarium specimens, we know that the radius of a circle and its area are related exponentially. So as the radius increases, the area increases exponentially. And of course, the area is occupied by bisexual flowers. By contrast, if you look at the relationship between the radius of a circle and its circumference, that relationship is linear. And so, of course, the periphery, the circumference, is occupied by pistillate flowers. So we think the marked increase in bisexual flowers relative to pistillate flowers is allometrically controlled. But the higher packing density comes at a cost. So again, this is coincident with what Ruben Torreses and his colleagues found. With denser capitula comes smaller fruits. And in South African Helichrysum, gynomonoecious species have achenes which are significantly smaller by about 10%. So in summation, we present the first phylogenetically corrected analysis of architectural differences for these 2 alternative sexual systems in a single large genus. We show that the acquisition of pistillate flowers, so shifts from hermaphroditism to gynomonoecy, can be remarkably frequent, 9 times in this group, excluding the terminals, with losses of pistillate flowers being less common. So what this tells us is that, at least in this group, once Gynomonoecy is acquired, it tends to be maintained. And then perhaps the most striking finding, Gynomonoecious species produce about 3 times more flowers per unit capitulum diameter at the cost of achenes about 10% smaller. But what's not yet clear is whether the ovule to achene conversion rates between Gynomonoecious and hermaphroditic species are similar. And then finally, we see that evolutionary shifts to Gynomonoecy entail the addition of lots of new bisexual flowers and comparatively few pistillate flowers. And this is contrary, again, to theories around how Gynomonoecy evolves according to Darwin. Darwin actually discusses this in The Origin of Species. Bauer and Beach again discussed this in 1981. And the model they describe is one in which the peripheral bisexual flowers undergo androecial sterilization. So those peripheral flowers become female. That was their supposed model. But what we show instead is an entirely distinct model where shifts to Gynomonoecy entail the addition of lots of new bisexual flowers and relatively few peripheral pistillate flowers. So I thank my collaborators and thank you for listening. And we welcome any changes. This work is in preparation, so this is the best time to get questions. Thank you.
Thank you so much for such a great talk. It's similar to our previous speaker. If anyone has any questions, you're welcome to unmute yourself and to be able to ask them. Otherwise, you can put them in the chat and we can read it for you. Okay, so Bruce Baldwin has a question from the chat that I'll read out. Any thoughts on the possibility that marginal pistillate flowers provide a brief period of head unisexuality, thereby enforcing outcrossing and limiting self-pollen stigmatic contamination?
Yeah, so this is a theory that I didn't mention, but is written into the paper. This is another considered benefit of Gynomonoecy, is that because these flowers lack stamens, they undergo less pollen clogging with self-pollen. I think that's what you're alluding to. Is the suggestion that these peripheral flowers are temporarily bisexual? I didn't quite catch that part. Maybe I should read the question.
I was going to say, I think he clarified that the heads are temporarily unisexual.
Oh, I see. So the question is, are these flowers, when all the other flowers are still in the bud stage,
yeah, that's right.
do they provide temporary, essentially complete femaleness?
Right. That's all I was
Right.
bringing up.
Um,
Just that there might be a brief period, where you don't have stigmatic contamination by
right.
self-pollen. I mean, there's still overlap oftentimes when pistillate flowers open relative to staminate flowers, but having them commonly being marginal florets, the unisexual ones, sort of lends to the question of whether that might, offer a brief period where you could have this unisexuality that would tend to enforce, pollen coming from outside the head.
Yeah,
Which
I
could come
haven't
from another head on the same plant, of course, but it's at least a brief period.
I haven't looked at very many species of Helichrysum, but those fresh heads I have dissected, the pistillate flowers don't often form a complete concentric ring at the periphery. They're often interrupted by bisexual flowers in peripheral positions. And what I recall observing is that the concentric rings mature in sequence anyway. So you don't have just the pistillate flowers maturing at once. There are entire rings of mixed sex maturing simultaneously. But that might differ in species where you have complete rings of pistillate flowers.
Right. I was thinking more with a lot of radiate heads.
Thank you.
Sure. Thank you. It's a great talk.
Thanks.
Andreas, if you want to ask your question.
Yeah. Thank you, Zaynab, for your talk. It was interesting. I was curious. I don't know anything about this group, but I saw that it's like very diverse, and I was wondering if you like, since you're still working on that, if you're able to polarize the shape of the receptacle on the species, because I don't know if there is variation in the shape on the receptacle, and having these ratios on the diameter maybe are altering a little bit the number of flowers that are being counted in the unit. So it's different if the receptacle is flattened and that's a diameter, but there might be some species that have like some physiognomy on that is giving you major numbers of flowers per the unit that you're analyzing. So I don't know if that's happening. There might be some Gynomonoecy's capitula that might have more flowers because of the shape of
Yeah,
the
precisely.
unit.
I see what you're getting at. You're suggesting that there might be some which are flattened and others which are maybe slightly conical, and that's how they're housing more flowers. The Helichrysum, again, I haven't looked at all 245 species of the South African Helichrysum, but those for which I've done receptacle measurements. Hela's work was somewhat incomplete, so she doesn't have measurements across the board, and where there were gaps, I did the measuring. And certainly from dried specimens, the only receptacle type I've seen are completely flattened. And they're usually approximating a circle. I haven't seen any aberrant shapes.
Thank you.
Thanks.
Okay, I think that's all for today. So let's thank our speakers again for giving their talks. Oh, I think say there might be a question in the chat if you guys want to catch up on that. But otherwise, that's all that we have for today. So if you want to see the talks, they'll be on the YouTube soon. Otherwise, we'll see you next month. Thank you.